In an earlier video, we referenced the website Uniprot. In this video, we'll go through the functions of Uniprot and of the website ProtParam and when these are useful. Like RCSB, Uniprot is a publicly available database. Uniprot differs from RCSB because it has one entry per gene instead of one entry per published structure. Proteins registered in Uniprot will have information from several different databases and publications. The RCSB database is best when you seek information about specific published structures, and Uniprot is ideal when you want more general information about the protein. On the Uniprot front page, there's a search bar at the top of the page to the right. Underneath, there's another bar which shows different ways of searching in Uniprot. BLAST and Align will be introduced in more detail in later lectures on bioinformatics. In short, BLAST is a search method that takes a nucleotide or protein sequence and finds sequences in the database that are similar, and Align compares two or more sequences. Retrieve/ID mapping searches can search by several identifiers at once. You'll not have to use this search method in this course. Peptide search searches by exact protein sequences. SPRQL is a more complicated search method that is also not relevant for this course. The most important search types to know is regular search and peptide search. When you use the regular search, you can search by common English names just as you did in the RCSB search. We want to search for carbonic anhydrase, so we type this into the search bar. There are two categories of entries: Swiss Pro, which has been reviewed by a curator, and Tremble, which is information that has not yet been verified. As you can see, there are substantially more unreviewed than reviewed entries. To the left in the search results, you can see their accession number and entry name. These are identifiers in Uniprot. An identifier is an expression that, when searched, redirects you straight into the corresponding entry. Accession numbers are codes of about 6 to 10 symbols that can either be numbers or letters. Entry names consist of an abbreviation of the protein name, followed by an abbreviation of the organism the gene comes from. If you click on the accession number, you enter the entry for carbonic anhydrase 6. Here we see a high annotation score, meaning that the protein is well annotated and there's a lot of information available. To the left, you see several subsections, which we'll go through one at a time. Under function, you find most of the biological information, such as general description, information about catalytic activity, possible cofactors, important amino acids for active site, ligand and ion interactions, and relevant metabolic pathways. It also contains information about biochemical properties, for example, kinetic parameters. For carbonic anhydrase 6, or Ca6, we see that the catalytic activity is related to transformation of bicarbonate to CO2 with the cofactor zinc. There's information about some inhibitors, and under sites we see that residue number 85 is important for the active site. The yellow bars are all references citing where this information came from. Under names and taxonomy, you can see different synonyms for the protein name and the name of the gene that the protein is transcribed and translated from. Here you can also find information about the genomic placement and the assumed evolution of the organism. Under subcellular location, you can see where you find the protein, indicated by yellow. Here we can see that the Ca6 is found extracellularly because it's a secreted protein. The section Pathology and Biotech contains information about disease-related mutations and any drugs that have influence on the protein's activity. PTM, or post-translational modification and processing, contains information about possible modifications of the protein. This can, for example, be glycosylation sites, phosphorylation sites, or placement of the signal sequence. The expression section has information about the gene expression on both mRNA and protein levels in different tissues and at different developmental stages. Here you can also find any effectors of expression, such as activators or inhibitors. Interaction has information about quaternary structure and possible binding to other macromolecules. The structure section, like RCSB, show the protein with the program JSMOL. There's often several structures, but in the case of CA6, there's a single structure with a PDB ID 3FE4 determined by X-ray crystallography. A new addition to Uniprot is the AlphaFold structure. AlphaFold is an artificial intelligence program that is recognized for calculating predicted structures with high precision. Therefore, this is not a structure determined by experimental method, but a purely computational prediction. If you look at the predicted structure for CA6, you can see that it's colored according to how certain the program is about the calculated precision of a given area. The structure section also has a little bar showing the secondary structure. Under Show More Details, you find a table showing the exact sequences with the given secondary structure. The section Family and Domains contains information on domains and motifs found in the protein, but also which protein family the protein belongs to. For now, we skip the sequence section and go to the section on similar proteins. This section contains links to other proteins in the database, which has either 100, 90, or 50% sequence similarity with CA6. Here you often find proteins in the same protein family. The last section we'll look at is the sequence section. This section shows full translated protein sequences. Here you can also find information on possible isoforms. Under experimental info, you see common variations that differ from the canonical sequence. Natural variants are caused by population polymorphism, and alternative sequences are sequences found in different isoforms, for example due to alternative splicing. If we return to the sequence, there's a little bar on the right side. These are direct links to programs that can process the sequence. This is, for example, ProtParam or ComputePI/MW. ComputePI/MW calculates the molecular weight and isoelectric point of the protein. ProtParam calculates both of these and more, so let's take a closer look at ProtParam. When you first open the page, you have the option to limit your calculation to a particular fragment. We're interested in the whole protein, so we just click Submit. Now we can see information on molecular weight, isoelectric point, amino acid composition, and extinction coefficient. There's also information on the expected half-time, stability, and hydrophobicity of the protein. As you can see, you can relatively easily obtain a lot of information from both Uniprot and ProtParam, which can be used for structural analysis.